morning, everyone. We're going to go through a bunch of examples of how to use the command line for various things. I think this was initially set up, broken into sort of two sessions. First was exploring data from the database in the site, and another one on looking at your workspace data and running applications. So we'll start out with the initial one of going through the data in the site. The information here is that we're back in the usual BV-BRC workshop space here under Command Line Interface. For this one, we've got an HTML version of the slides that I'll be going through, and you should be able to copy and paste the examples right out of the slide into your command line if you want to follow along. I've done the copy and paste on, I think, all of these. I've done a little editing, so I may have screwed something up, but hopefully it'll all go. So first, we're going to talk a little bit about where to find more help on these things. At this point, it's a common story that under the help menu at the site, you'll find there's command line interface docs. This link should be live. It will take you over here. We've got a notes on installation. We've got some reference documentation. We've got a set of tutorials. We've done a sweep of changing things from Patrick to BRC through here. You may see some icons and some examples that still have Patrick in them that all should work the same. The tools are all called P3-something standing for Patrick 3. We're not going to change that because that's all wired into what everyone's doing. And so it's now just think of it as an abstract token that the tools start with P3 and you don't need to worry about what that used to stand for. Also in that main tutorial, there's a command line reference. This has a link to documentation on each of the commands that we'll be looking at. Some of them have, more or less information in them. That we've got some to-dos in here. If you smack into something that you can't understand and we don't have anything in the documentation for it, send us a ticket. We'll get that filled in. As with a lot of things, the detailed documentation sort of lags behind the other things that are going on in the project. Also, each of the commands has a help option on it that should give you a sort of more or less useful help message. And again, if you're trying to do something and you can't figure out from the help message in the command what it is you're trying to do, shoot us a note. We'll try and get all this cleaned up. A lot of the command line stuff are things that we use internally, and so we know what they all do for the most part. So if things are confusing, let us know, and we'll try and get things cleared up some. We all know how to install the CLI. We went through that yesterday successfully, hopefully. And we did a little of this yesterday, too, because we have authenticated data in the database. You need to log in to be able to see either your private data or your workspace data. This is the P3 login command that will look for your BV-BRC password. It'll tell you you're logged in. There's a command called Who Am I that will tell you whether or not you're currently logged in and what your login is that you're using. The way the logins work is that there's a file that gets written into your home directory that's a authentication token for getting to the resource. It's got a pretty long expiration on it, but sometimes you'll try to run a command and it says, oh, your token has expired. That means you need to run the login again. I'm going to talk a little bit about how the data is set up in the database, because that affects how you think about using some of the commands. If you're a database geek, we think about these things as an entity relationship database where we have entities that are storing, sort of the things that we're interested in, like genomes or proteins or drugs. And we store also the relationships between them. In the diagram here, the squares are the entities and the diamonds are relationships. So here at top middle red box is the genome. That's sort of the center of everything. Everything's linked to a genome. Off to the right, we've got the entity of drug, which is related to by this entity that says, we've got this resistance data with this genome. Off to the left, we have the contigs, DNA sequences for the genomes that are linked to there. Moving down, we've got a relationship between the genome and the features on the genome. When we say feature, what a feature is in the model is it's a sequence of an area on the DNA sequence of the contigs of the genome. It may have spaces in the middle of it. The notation supports introns, and we have many types of features, and they can have associated protein translations for the protein features. Also, then features are grouped into families. These are the PGFs and PLFs, protein families we saw in the interface. And so we can explore those as well. So there's a common model to how all of these commands work. This is very much driven by the philosophy that comes way back out of the origins of Unix, where you store all your data in flat files. We have tabular files, and pretty much all the commands operate by taking in a tabular file, doing some processing or data lookup, and spitting out a new tabular file with additional data or change data. So if we have a file here that's a table of genome IDs and genome names, this file has a header that tells you what these columns mean, and it's got the data in them. We can run some commands, say p3 get genome drugs. This is what all of our commands are going to look like. We can give it a column that we're going to operate on. So here we're saying I want to look at the genome ID column, and I want to extract some attributes from this genome drug relationship table. So I want to get the resistant phenotype in the antibiotic that's resistant. So when I run this command on this input data, I get added columns of resistant phenotype and antibiotic. So I take an input data set, I'll look up against the database, and I've got an extended data set on the output. So these scripts have a set of common notions that you'll see across the board. Again, they all operate on tab delimited files with header lines. The scripts are designed to be piped together. If you're not familiar with using the pipes at the shell, these are shown using this vertical bar character. And some common options you'll see is this -col to say what we call the key column that tells it what data we're operating on. Say no head if you've got a file that doesn't have any headers on it. If you forget to use that and you've got data that doesn't have headers, it'll think the first line of your data is a header line, and get possibly goofy results, and you'll drop the first line essentially. -help will show you the help message. -fields is useful in any of the things that let you select data based on available fields in the database. This will dump out all the fields that are acceptable there. I use this all the time to remember. In the genome table, what do we call the genome name? Is it genome name or scientific name? Here, you dump it out and you search and say, okay, it's a genome name here. A couple notes on running the examples here. Like I said, you should be able to copy and paste out of this document into the command line on your computer. Some of these have very long pipelines that span multiple lines. In this case, the / character will show where the lines are split. If you're on a Mac or a Linux machine, you can just copy and paste the whole shebang, and the / does the right thing. If you're using on Windows, it doesn't understand the backslashes. So you'll have to copy and paste the meat of each line separately and paste it into a single long line on the computer. We have some additional common parameters for the data query scripts. A number of them support filtering, and can filter using the usual relational operators, equal, less than equal, and so on. You can pick additional attributes to add to the output, and you do this with the -attribute and then a list of names. So for instance, if you wanted to query the database for all genomes that have a genome name of Staphylococcus aureus and that have a genome length longer than 4 megabases and retrieve the genome ID name and taxon ID, this would be the command here. I'm just going to copy this and see if I can get this to start up. Let's see if it's going to cooperate. I have to go over here. There. It's not really an application. It's a wrapper that sets up the terminal application with the right environment to be able to get at the commands. The Windows one is similar. It's an application icon, and it will just start up a terminal window with the environment set up. So I'm here with this. So here's my command that I had copied out of there. We can see now that these P3 all commands are a great place to start a pipeline, right? Because I've posed some question. I don't have any data right now. I posed a question with the command. I said, give me all the genomes with these properties. And now I have a nice tab delimited file with headers. So I've got my genome ID, genome name, taxon ID, along with the data here.
About where it says genome.genome ID, just the genome is actually the data store that it's pointing to or database, right?
Right. If you do P3 all genomes without any query limits, it's going to try and download, 7 million records. The one thing that I want to comment, though, that we do a lot of running a thing and saving the results in a file. Say that we've done this query and we want to start, working with this data. I want to put it in a file. See, I'm going to make a directory here on this loaner laptop for the workshop. To do this sort of things, if you haven't used the command line at all before, there's a ton of tutorials online for like a basic Linux tutorial that will tell you what it means to make a directory at the command line and move around the file system. I'm not necessarily going to go deep into that. Also, if you're new to using the command line, the arrow keys will probably let you go back and forth through the command history, so you don't have to copy and paste again if you're just going to rerun a command. So I'm going to arrow key up here. We had someone that ran into the problem with the command not working on Windows because of backslash not working right. Let me show you what I mean that you have to do instead. So this command is on 2 lines here. If I'm on Windows, I'm going to have to do it like this. I'm going to copy this first line up to everything just before that backslash. And so I'm going to copy, I'm going to paste it in here. So now I've got the first part of the command in my command window. I'm going to have to come back here. I'm going to copy the second part of it. And then I'll paste it at the end of the command line here. I've done what I did before. It just took 2 copy and paste to do this. So now I've run this. I want to use this as an input to some other analysis without rerunning this command, because some of these commands may be quite expensive, so you don't want to run them more than once. So I'm going to take this command that I ran. I'm going to use the greater than sign with the spaces around it, and I'm going to give it a file name. So I'm going to call these longstaff.txt. All right. So I ran this command. It didn't show me the output. If I do an ls here, see, I've got a new file called longstaff.txt. I can cat the file out and I've got my results. So now I've got a data file I can do something with. So we're going to walk through how to explore data and the types of data we have in the database. We'll start with the genomes. In the BV-BRC is a data object that holds the DNA sequence for the genome. It holds any metadata that's associated with the genome. I'll show you exactly what we're storing, because it's anything that we see at the command line. This is all data that's the same data that is driving the website. So that any of the things that we see here in the genome overview is data that we can query via the command line. So when we were looking at genome name equals Staph aureus, what we're doing is querying the same underlying data that populates this genome name field here in the GUI. When we're saying genome length greater than 4 megabases, that's querying the same data source that fills out this genome length field here. And so if you're trying to figure out how to do something in the command line and it's not clear, what I would do is come over to the website, sort of browse to the data I'm trying to get at in the website, and then you can translate that over to a command line query. And again, if you're stuck and got something you can do on the website and you can't figure it out on the command line, let us know. We're happy to help with that. And talk a little bit about the identifiers here. We've seen genome IDs, and that these are these number.number. The first number in the pair is the taxon ID of that genome. A subtle point, but this comes up sometimes, that's the taxon ID that the genome had when we loaded it into the database. We use the NCBI taxonomy to name these things, and they shuffle stuff around, right? In the last couple of years, we've had species names change and the like. We don't change the identifier for the genomes in the database, because that way leads insanity, because we want people to be able to write a paper and quote a genome ID, or have a data file and have genome IDs in it, and have, once that ID is been given, that it's not going to change. The second number in the genome ID is just an index. For every new Buchnera that we put in, we're going to have 9.x, and we'll just have 9.y. If we have 9.10, then we assign 9.11 for the next one. There's almost no meaning in the number after the genome ID. It's just an index of how many genomes of that taxon we've ever installed. In this system or any other system that share that genome ID is actually, if you're a RAST user, we guarantee that if you annotate a genome in RAS and annotate that same genome in BV-BRC, you're going to get distinct genome IDs. You're not going to ever run into a problem where you've got 2 genomes with the same genome IDs. Bacterial genomes that have multiple contigs will all be assigned the same genome ID. Currently, with the viral genomes, each segment in the viral genome will get a distinct genome ID. So they'll have a genome with a single DNA sequence, which is that segment. This may or may not change, but right now, that's the way that the virus data is stored. And starting with the genome is often a good way to start working through a problem. You can use the P3L genomes to list or query the genomes, like we saw in the example we just ran. Given a genome, you can follow down to the features of the genome by using get genome features. We can follow to the DNA sequences by using get genome contigs. And you can go through the drug resistance data by going through the get genome drugs. So this takes, remember, we've got this sort of cross-shaped data map. These are the commands that will take you, left, right, and down to access the data. We'll talk about features a little bit. So a feature, again, is a region on the genome sequence. It's defined by the genome and sequence identifiers plus a set of regions. Feature identifiers have this more complex form. They start with fig bar, the genome ID. So this is this, number format that we just saw. A feature type, which is a short or medium length string, like PEG for protein encoding genes, RNA for RNAs, and so on. And an index. The index is the index of the feature along the chromosome in the genome. We start at the left and assign left to right for each feature type. Each feature type starts at 1. So you may have 1 to 3,000 for your protein features. You may have 1 to 6 for repeat regions, and so on. So some useful programs for exploring the features are, again, get genome features. We're given a genome ID. We'll enumerate the features in the genome. Get feature data. If you've got a file with feature IDs, that will let you retrieve additional metadata. Again, the metadata there is everything you can see in the feature page on the website. And you can use get feature sequence to actually pull the DNA or protein sequence, and you can get that data in FASTA format, so that lets you invoke other analysis tools. We've got a tool that exposes some of the text search mechanisms called P3 Find Features that will let you search for features for things like locus tags or protein IDs from RefSeq, gene names. If you've got the MD5 signature of the translation, there's a number of places in the system where we are working on with protein translations, but we don't want to work with the entire long string. So we compute what's called the MD5 signature. This is a short hash of the protein sequence that is going to be unique across all of the protein sequences in the database. So we use that as a proxy for the actual protein sequence. We'll see it's an example coming up. And the product will let you do a search against the functional assignments for the features. The drug tables that you access are antibiotic data. You can use P3 all drugs to get a list of the drugs. This is one of the all things that's actually should run quickly. We'll see what we're talking about if we run this. So these are all essentially antibiotics that we've got AMR data for. All right, and you can get P3 drug genomes to get the list of genomes given a drug name. We have some other entry points that are useful for exploring the data. You can get all the taxonomy categories using the P3all taxonomies. If you're doing subsystem analysis, we have queries into getting the subsystems and subsystem role names. You can also look at the genome sequences using the all contigs. Remember that these P3 alls really are all if you don't specify any queries on them. So P3 all contig, it's not going to return the contig data. It's going to return metadata about the contigs, but you're going to get probably tens of millions of records if you let that run long enough, which you can if you like. These tools are set up so that it may take a long time to run, but the queries are chunked. And so hopefully you won't get timeouts. You'll just be spending your time waiting for it. The system should absorb a fair bit of traffic of these sort of things. That said, if you're running something and every time you run it, the website crashes, let us know. That's not supposed to happen. And some of these expensive queries are useful, and we'd be interested in knowing what's the big data that you're doing that's causing problems, and we can figure it out a more optimized way to take care of it. We've got some utility scripts for dealing with these files with the headers on them. People are using Excel. Some of the things you do in Excel with tabular data too, right? You can extract and reorder specific columns using P3 extract. You can sort by column. Using P3 match, you can extract data based on conditions of the values in the columns. You can join files together on a key field. If you got two files that all have genome IDs in it, you want to merge that data on genome ID, you can use P3 join for that. You can use P3 head to just look at the first few lines of a file. If you're trying to explore what's there, you can use P3 echo to create a little file. We've got an example of that. We'll see in a minute. In fact, we'll have it right here. Remember, we talked about needing to get sort of a starting point for one of these pipelines of commands. The P3 all commands are one way to do it. But if you know that you have a particular set of data items you're looking at, you can use P3 echo to create one of these data files to start with. So say I want to start with a couple of genome IDs that I know. I can use this P3 echo. I can tell it the column name I want is called ID, and I'm going to give it these two genome IDs. So we can try this example. I'll just copy and paste just the command. So I'm going to put that there. And so I just get this very simple data file that's got a column name of ID and 2 values. If I wanted to say, I want this genome ID instead, I can come over here, call it genome ID. So say I want to save this in a data file for use for later. I can make a file called starting genomes.txt. Now I've got this starting genomes. Now I've got a little file I can use to bootstrap some further analysis if I want to. So what we did was we just created a single column table. If you use that -t flag twice, we can get a 2-column table. So say we want to have an ID and a count. So anyway, we're going to make a little data file that's got genome IDs and a count field on it. So I'm going to say P3 echo. I've got one column that's going to be genome ID. And another column I'm going to call count. All right, so now I've got a 2-column table. Now I'm going to give it some data values. Give it that ID and say 46. Now what this has given me is exactly that. I've got a file with a headers with my genome ID and count and a couple data values. And I can send this into another program. So we're going to solve a little bit of a real-life problem here. How many proteins have a protein identical to this given protein identifier? So this is the CoA protein and strep mutans. And so the way we're going to solve this is knowing what I mentioned before is that for all of the features we store in the database, the MD5 is checksum for the protein translation. So we're going to find all the features in the database that have that protein translation, and we're going to count the genome IDs. So we're going to start out by figuring out what is this checksum value for our feature. So to do that, we're using our P3 echo. You notice we didn't give it a column name option here. And if you don't give it any column name, it'll just give you a default. So let's just run this P3 echo without a -t and see what we get. So we get just a name of ID. That's fine. That column name is just for our use in working through these things. Then we're going to use P3 get feature data, which given feature IDs as input, it will pull additional attributes for those features. And so what we're going to pull is the attribute called AA sequence MD5. And say, well, where did that come from? Remember I mentioned we've got this fields option. There's a get feature data.
What do you want, Bob? Identical proteins or identical genes?
I want the number of genomes that have that protein.
Okay. The identical sequence?
That have proteins with that identical sequence.
Okay. I've got 113.
Okay, we'll see.
I haven't sorted them to see if there are duplications.
Okay.
Genome. Are you going to do that?
Yep.
All right.
Yep.
The race is on.
All right.
Come on.
Also, if you're on Linux or Mac, you probably have tab completion on the command line. So I can say, if I say P3 get and hit tab, this will expand out and show me the possibilities for what the P3 get commands are. It's just nice for, if you don't remember quite what it's called, you type the first part of the command in and it will expand out. And what it does looks a little different depending on which operating system you're on. Anyway, so I was looking for get feature data. And the fields flag.
I've got my answer.
All right, we'll see if we match.
Yeah.
This dumped out a bunch of stuff. That's because we have a lot of fields available on the feature data. So we've got amino acid length, sequence MD5, which is the one we're looking for. Accession. If you went and looked at the feature page, these would all look very familiar to you. Some of these things have this keyword related in it. That means that that data is actually coming from a related table out of the database. For instance, in the database, we store pathways, somewhere other than in the feature table. But this script will go off and find that data for you. It will probably make it a more expensive search because it has to look up in multiple places for the data. Some of these also have a tag called multi. And what that means is that these are multi-valued fields. And the scripts have a way of encoding these multi-valued fields that I think usually defaults to comma separating them. But I think there's also command line options. You can change how you do that lookup. We're going to use the AA sequence MD5 to pull out that sequence MD5 attribute. There we go. There's a bit of a pause there. That data was probably, not in memory on somewhere in the database and had to go pull it off a disk. But then we've got this long hexadecimal string, which is the hash value of that protein sequence in the database. say we want to see what that sequence is just because we're curious. If I go back up to this field list here, seems like I should be able to find that. So let's scroll through here a little bit. We got the amino acid sequence. And this is also tagged as related. This is because the amino acid sequences are highly repeated across the database. There are many, many proteins that are in many genomes, right? And it's a big waste of space to store the full translation for every feature in the database. We've got thousands upon thousands of E. coli's that are nearly identical. And it's a lot of repeated data. So we store those off in another table. So this related thing says that this query is going to go off to that related table. So just for grins, I want to look at the sequence length and the sequence data for this guy. So I'm just going to add on a couple attributes here. AA sequence. You notice that I don't have a length in here. And that usually means that I misspelled it. We've got sequence MD5, sequence length, sequence, and in my data, I've got an MD5, nothing, and my sequence. What that means is I misspelled the field name and it didn't find anything. So I'm going to go back and use a length here. Now I can see that this is a 306 amino acid sequence. The other thing to note is that if you're exploring like this, I do a lot of fiddling, running commands, see what the output is, run it again, tweak the inputs a little bit. Gives you a feel for how to play with the data and see what's there. You really can't break anything. You may do something that runs for 10 minutes and gives you humongous output. And then you learn how to not do that because it's easier to pull smaller amounts of data. Those queries will come back really quickly. We've run our command. We've got our sequence MD5. This is another common problem. So he ran this part of the command here. So if you run that, it'll just sit there. And the reason it's just sitting there is it's expecting to get input to tell it what feature to look up, right? And so what it's doing is waiting for you to type in that data.
That's the difference between get something data versus get all.
Right. P3 all doesn't expect any input and it'll just spit it out. The gets expect to get something in. So if you're running something that it thinks should be running fast and it just sits there, you may have made that mistake. That's another real common mistake to make. Okay. Remember, our plan here was to find all the proteins that have the same checksum and count the genome IDs. So now we've got our checksum, right? I'll rerun this short command that just gives us what we want. So we've got our checksum here. So now we want to find the features that have that checksum. And if you recall, we just talked about a command that will help you find features. And the AA sequence MD5 was one of the types of features you could look for. The first part of the command is the same. We're going to use P3 echo to create a data file that's got our ID. We're going to use get feature data to pull out that sequence ID like we just had. And we're going to pipe that into P3 find features, which is our search command. We're going to have it search for the sequence MD5. We're going to give it the count flag that says, I'm not interested in all of these features. I just want to count how many there are. I got my command from the slide. Okay. And this last step in the pipeline is done what these other ones have done. Our first two columns are the ones that we saw when we ran the get sequence MD5. Let me make a little space here so we can see what's going on. We looked up the sequence MD5. Then we did the find features, and what we spit out are identifier, our MD5, and this count. That's 315. Oh, and Rebecca's left.
Let me tell you her results. She found 207 identical proteins.
So these numbers are different. As we say here, this is not the right answer. This is the total number of features that have that protein,
Right.
not the number of genomes, because we have genomes that have multiple copies of this protein. So what we want to do is count the genomes that have the features. We're going to use the same basic pipeline. And on the find features, instead of just getting the count of features, we're going to use find features with attribute genome ID to pull out the genome ID for those features. And then we're going to pass it through this utility called P3 count that we talked about a little bit before, and that we're going to have it count the genome ID field. So I'm going to run this command without the count at first, so we can see what this output looks like. This is another thing I do all the time. You run little bits of the pipeline to see if the data that comes out are what I'm expecting. So I'm going to run the echo to the get feature data to the find features. So now we can see that I've tacked on this column here that we've got. This is our initial query feature. This is the MD5 of it, and I've tacked on the genome ID of the genome that we found us in. So now what we're looking for is the unique values in this last column, which is what tacking on P3 count does. And we got 207.
Yes.
All right.
So
So the world is consistent.
at least the database hasn't changed enough.
Database hasn't changed, and she apparently doesn't have any private genomes that contain that feature. This is another place where you'll see count discrepancies, that if you're looking at somebody's private data, because the private data is rolled into the database with everything else, that will affect those counts. That also means I also don't have any private genomes that have that protein in them.
This is deriving based on sequence, exact match of sequence.
This would be the exact match of the protein translation.
So the other little interesting fact here is that the multiple copies here, the reason you have 315 genes and 207 genomes is that we have the same feature annotated by GenBank and the same feature annotated by the Patrick annotation system. So it's not that you have a duplicate copy of the same gene in the genome, but it's that you have this exactly same gene annotated by the two different sources.
In this case, I'm not sure that's it.
That is it.
Well, let's see.
You can filter
Well,
by,
let's see. We can
filter by annotation type and you can check the number.
yeah. So let's look at the features we got back on the find.
And I got it before Bob, right, Alan?
Yes, you did, but
Did you have to use Excel?
to be fair, Rob.
Did I have to? Did I get to? And the answer is yes.
All right, so I'm going to add on, in addition to the genome ID, as Malik was saying, perhaps this came because we had features that were annotated by other sources. I'm going to go back up to my list of fields here, and I believe one of these fields will tell me where the annotation came from. Let's see, I'm going to make... I'm going to pull the genome ID, I'm going to pull the annotation, and I'm going to pull the Patrick ID also. So again, I'm looking for all of the features that have that same MD5, and I'm going to pull out some additional information. And so what we're seeing here is that these are all Patrick annotations. The question is, if you were on Mac or Linux, if you'd be able to take these and pipe these into other scripting languages or analysis programs, and definitely. And even on Windows, if you've got Windows tools, people write Python code on Windows, you could pipe it into there. Since these are tab separated data, if you're doing machine learning work and you've got pandas data frames you want to populate, you can make data frames out of these, very easily. Yeah, that's all very smooth. So I guess the question to Malik is, how do we get our RefSeq versions of these?
Count it. How many you have?
The count there, that was our earlier, we got 209 features here.
So you already brought it down.
Yeah, that's a different number than we had before.
It's two more than it was before.
So didn't we get 315 from one of these? I'm going to rerun this. That's sort of interesting. This is where we need to go bother Bruce and ask him why these numbers are different. And it's not immediately obvious to me.
If I were doing it on the website, which I am, I can see the exception numbers for RefSeq right next to those.
Oh, I can ask for those.
BRC.
It's at RefSeq. And again, because I can't remember it, I'm going to go back to my gene list and the field list in here.
RefSeq locus tag.
RefSeq locus tag. Yeah. Here's our locus tags. Not everything has a locus tag on it.
Is that a feature that we've annotated that wasn't in RefSeq?
Yeah,
When we annotate a genome that comes out of RefSeq, remember what Jim told us yesterday, that we do all our own protein calling using Prodigal and Glimmer, and we do our own annotation. We also transfer over as much as possible the information out of the original GenBank calls for those features. So if we call the feature that's in the same location as the GenBank feature, we'll bring over all the RefSeq attributes like the locus tag and gene ID and the like.
When you say in the same location, do you mean the same start?
It's the same stop and strand
Okay.
and config. Sidetracking a little more. The other thing we do that Jim didn't mention is that when we're importing GenBank data, and we do the prodigal and glimmer calls, we also bring in all of the original GenBank gene calls. So we've got this big pool of all the original GenBank calls, plus all the calls that we've made. And we've got a genetic algorithm-based tools that basically goes in and sorts out the multiple overlapping calls. So we end up with single protein calls that are judged to be the best call, plus all of the other miscellaneous features that the GenBank organisms will have on them. So we don't lose anything by doing our own annotation. So it's just a short review of the pipeline. We use Echo to give us a starting point. We use the get feature data to get the protein identifier. We use find features to find all the features with that. And we use count to count the number of distinct genome IDs. To be fair, the other thing that you can do easily with these, I'm going to save this last output to a file. This is actually the thing I do all the time, at least once a month with monthly report data, is I can say, open in Microsoft Excel features.txt. Is this it? Yeah, this is it. This is that data that we just pulled out in the CLI in a form that the biologists can use now.
Let me tell you a quick story. The first time I went to work with these guys, and I mentioned that I liked Excel, and I was in a room with them, and Ross Overbeek, the god of our group, he said, "Oh, I hate Excel. I'm going to show you how to do better than Excel." And he was projecting onto the screen so you could see him coding. And I felt like I was getting shot with a machine gun. And then he said, "There it is. Isn't that better than Excel?" Looking at it there. And I'm like, yeah, yes.
So that wraps up the first little example. We're going to talk about groups now. So the other thing that it's nice to be able to do is just jump back and forth between working in the website and working at the command line. So you may do some analysis on the website using protein family sorter, say, or using, I've got a set of bins that came out of a binning run and it made a genome group for me. I want to do some work on the command line using that. There are commands for manipulating these groups. Say we want to get a list of all the penicillin-resistant strep genomes. So we're going to play a little bit with the resistance data here. I'm going to start a data file with the drug name of penicillin. I'm going to say get drug genome, so this will pull the genomes that have resistance data for penicillin. I'm going to say I'm only interested in strep pneumoniae. If we had a lot of resistance data, we could get a very large result, so we're going to reduce it down to what we're really interested in. We say I only want the ones that are tagged as resistant, and I'm going to pull out the genome ID and the genome name. So I'm just going to run this part of the command at first so we can see what it generates. There's quite a few here. Trying to get to the top of the command so you can see the headers. So we've got an ID column that we started out with, and we ask for genome ID and genome name. So here's our genome ID and our genome name. We've got a big list of them. So what I'm going to do is now use this command called p3 put genome group. This expects to get a list of genome IDs because that's what's in a genome group. And so I'm going to tell it that the genome ID in my input data is in the column called genome ID. And I'm going to create a group called strep.resist. So it didn't generate any output because the output went into the genome group on the site. Then we can now use get genome group to pull that data back out. And so there's our list of IDs. So now if I go over into BV-BRC, now if I go over here in workspaces and go to genome groups, I'm going to order by most recently created. I've got a genome group called strep resist. It's got 4,000 genomes. That makes sense because that was a big list that we scrolled through there. I can click on that and go over and click on view. Here's my summary data of this resistance of the set of genomes. I can look at the drug resistance data. I can look at this stuff. If I want to go do further processing them now, I can go with the genomes list. And now, so I'm now launched back into the website with a set of data that I've selected over on the command line. You could see you could do some analysis in Excel and write it out to a file and then load it in. It's a little fiddly. You have to tell Excel to write a tab delimited files. We'll leave that aside for the moment. You can also, there's a list genome groups command. So this will dump out the set of the genome groups that I have available if you needed to look back and see what you had if you couldn't remember the name.
It doesn't care about what directory or folder or whatever it might be in, or does it?
The question is, if I do the list, does it care what directory it's in? Right now, these will pull out of the genome groups that are in this genome groups folder in your home. That's the list that these work from. That's actually one of the things that's on our mental list to fix up, is to be able to give it a path name to a genome group.
So the command already has knowledge of that genome group.
Right. The command line is you can look at the genome groups in the folder called genome groups in your home workspace. What we haven't talked about really is that all the times so far that you've created genome groups and feature groups, they're all going into this either genome groups or feature groups folder in your home workspace. You can actually store those groups anywhere in the workspace. For instance, if you're working with someone else, you could stash a genome group in a shared workspace in order to look at it. Right now, command line doesn't know about those. It assumes that everything's in the genome group folder. It's on the list to be able to give the command an arbitrary path to a group file in the workspace. So that's genome group. Feature groups work just the same. So here we want a group of all the hypothetical proteins in strep pneumoniae R6. So here we're using P3 echo to make an input file with that genome ID. We're going to get genome features to find the features that have a product that matches hypothetical protein. And we're going to pull the Patrick ID as the data we want out and create a feature group. Notice again that this Patrick ID is another place that the Patrick ID name shows up, because feature IDs are so deeply wired into everything we do. You know, it's likely that that name is going to stick as Patrick ID just for mundane considerations of being referred to in so many places. And we're going to create a feature group called SP hypo. Now I can get that feature group. We've got our list of features here. Now we can flip the page, go over here to my workspaces, feature groups. Again, I'm going to sort on created to bring this new one up to the top. So there's 400 features. You can go view the group, get the list, and lo and behold, there's all these features that are hypothetical protein. Notice that we've got one with a feature called hypothetical protein in cluster. Because the underlying database is Solr, what it's really doing is searching. It's not doing, for these sort of queries, it's not doing an exact match. So we're getting the things that contain hypothetical protein. This is sort of a lingering open issue. It can be useful to do it this way. It can be annoying that it does this. If you really want just hypothetical protein, you would need to do a post-processing step that would clip that out. If we go back here and remember that there's one of these utility scripts called P3 match. Let's just try that here. I'm going to do P3 match -help because I don't remember how it works. So let's see what the column header was if this get it. And maybe I really do need to tell it the column header. So it's SP hypo Patrick ID. Column match. Oops. I was combining my two commands here. So anyone spotted what I'm doing wrong? It just now occurred to me what I'm doing wrong here.
Not using Excel.
In this case, I don't think Excel would have helped,
Okay.
because what I was trying to do was match for things that only had a function of hypothetical protein, but I forgot to pull the function out to be able to see what the function was, right? Right here, it's staring in my face. This is the data that I was trying to match on hypothetical protein.
What is the SP where you have SP.hypo?
This is me thinking that, I didn't specify a column to match on. I was telling it that I want to match on the column called sp.hypo.patrickid, which is
it's actually the column.
That's the column name. I had scrolled up to find the column name.
Gotcha.
But I was thinking a step ahead. I don't have a function here. So let's go pull that out. So I'm going to get my feature group. I'm going to do P3 get feature data. And I'm going to get, I think it's product. And then I'm going to match. I don't know what the name of the column is going to be, but I know it's going to be the second column. So I'm going to say column 2. So the P3 match apparently either it uses the same fuzzy match or it's specified my column wrong. So now that I've got this, I can do that. So I'm going to tell it I want to match on the column called feature product. And it's still doing that. So either I'm doing something wrong or this is the defined behavior of the script. So I'm going to queue up another question to Bruce. But I say Bruce is a guy named Bruce Borello who worked through a lot of these underlying database and command line tools.
Case insensitive, and it's just looking for that substring anywhere.
Right. We've gone down a bit of a diversion here. So let's go back to what it was we just did, which was did a lookup for the IDs in the database for this genome that have hypothetical protein in the product name and created a group in it, which then we've gone back into over on the website and we can use the data there. And I believe with this, we come to the end of this part. Yeah, this was it here.